me, see the screen, see the pointer, everything. There, chat. So this the last lesson of today would be yes, I know that one. About protein domains. So what are protein domains and why it is important to know about that in bioinformatics and in general if you are analyzing proteins and protein structure. So protein domains are structural units. The average is 160 amino acids. In humans, don't learn this number. It's not important. Just it's important for you to know that that these units they are important because they tend to hold particular functions. So, you know, the protein as a global thing will have a general function, but the domains as subunits may have a particular function, like binding DNA, for example. So you can have a protein, it contains a domain that is DNA binding. That domain is doing the DNA binding, but then the entire protein itself is binding to DNA somehow. Okay. But they are also units of folding. Okay, so they will fold separately from one from the other. This maybe you can already. I'm already pointing here, so you can appreciate here. I just move to the next one. So this will be one domain, and this is another. If I follow the alpha chain of this protein, there is only one alpha chain, and it goes like, you know, here up, down, blah, 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 and then crosses only once to the other part, and then it here goes and makes some kind of globular thing. Okay, so there's only one cross. It's very unusual to have two domains and to have the alpha chain going up and down, you know, to the one or the other. And this is also due to the evolution of these domains. So these domains may evolve separately, may come from ancestral proteins that were not together. So this domain may come maybe in another species, maybe in a protein, and this domain in another species. You know, the similar protein maybe in a separated protein, and then in one particular species, this domain could be joined. Okay, so in evolution, these things happen. Domains may be reused, copied multiple times in the genome, and then reused in different proteins. And this, you may have a DNA binding domain that it will be copies will be made in evolution, and those will be used in different proteins, giving many proteins the ability to bind DNA, and these combinations give incredible functional properties to different proteins, and this is why this happens. So, and the number comes back again, and this is why it's here. The average protein is 300 amino acids, and the average size of domains is 160. So you can see that most proteins actually will have more than one domain, or multi-domain. So this is a feature that is extremely important. But there is a large range of variation. There are small domains, maybe with 60 amino acids, 50, and there are very large domains that have maybe 1,000 amino acids. So why are we worried about these domains? Yeah, don't learn this by heart. This is I'm not going to ask this, but there are methods that determine protein structure have size limitations. This we will see tomorrow with Elmar, which will speak about protein structure determination. And because these methods cannot deal with very large proteins, sometimes it's useful to chop these proteins. You know, you cut the protein in fragments, as we have seen before in the example we have been looking at. That was a fragment, and the resolution experimental resolution of the structure of a fragment is normally easier than solving the structure of the entire protein. Okay, so this is reason number one. The second is that when you want to find out if, for example, one of these DNA binding domains is present in many proteins, you can have bioinformatic programs that allow you to compare sequences and find those. And of course, these programs normally work better if you go domain by domain. Okay, and is much more powerful instead of looking for entire proteins. And this we will see on Monday, and this has become really like a big breakthrough. The methods that can learn to predict the structures, for example, and methods that can predict function of domains, which we will not see very much, work much better. So if you try to predict the function of an entire protein, it may not work very well, but when you go domain by domain, it's much better. So then you can see, this domain is a transmembrane domain. This is an RNA binding domain. This is a domain that phosphorylates another protein. And then you combine these things like Lego blocks, to build different proteins. And it's a beautiful way to gain protein functions in evolution. Now, because this bioinformatics, so there are databases of protein domains, so that you can, for example, find out the motivation is that if you have a protein sequence and you don't know the structure or don't know very much about the function, it's like a protein that you are studying and you want to know which domains this protein have. There are databases that allow you to do those predictions. Okay, and basically most of them work in the same way. And I'm going to introduce to give you an example of how bioinformatics work and how in bioinformatics sometimes we have several ways to do the same thing. Like I said, you have the program Chimera to represent protein structure, but there are other programs that represent protein structure, but they work more or less the same. We have several databases that will help you about finding domains in your proteins. Okay, so one of these is SMART, and what they do is they will get examples of a particular domain, then they will align them together. You will see this a bit maybe more on Monday. What does it mean? But basically, you can kind of learn from the sequences of some examples. You can make a kind of a profile, which then you can use to search in the database of sequences. Okay, no database of structures, the database of all the sequences, all the protein sequences. You can search for more domains, DNA binding domains of a particular type. Okay, and this they can do. I mean, they cannot do this automatically. You have to go domain by domain, taking some examples, generating a profile, using it to search for more, and so on. So it's some work, but it's quite worth it. And so, like I said, the details doesn't matter. But so if you go to this database, you get a window like this, and then you can query the database in different ways. So, for example, of course, it's a domain database, so you can interrogate about a particular domain. In this case, SH3. And so you get some text informing about what is known about this domain, what's it, some papers that talk about it. But also you can, like I said, ask about the domains that are predicted. Okay, so say that we are looking at a protein sequence for which we don't know the structure. Still, they can apply the profiles that they have generated. And then in this case, I'm interrogating this website with the identifier in a database, okay, of a particular protein, which is SORLA, is the human protein. So I put identifier there from a database, and then the program will return after a very little time, because they have already these things normally pre-calculated, this view where it's telling me there is a very large domain. It's very large because I have this scale here for the residues. So this is like a large domain there. Then there are 4 instances of a domain over here. You see it's much smaller. Then there is another here, and so on. Okay, and then it will, depending on the database, they will do other things like predict a transmembrane helix. There is, I think, a coil here. There is a coil. there is a signal peptide here. So depending on the database in particular, you will have other features that are calculated. And also to go on with this particular database, you could ask, okay, may I have other proteins in the database which have the same composition of domains as the one that I found already. So this was my then it finds this other protein and so on. Okay, so it will tell me other proteins if I want that also have the same types of domains. Okay, not necessarily in the same order. If you are interested for that. So, for example, regarding particular domains, you can always, like I said, ask for information on those. Find out in this particular database in which structures in the PDB they are used, if you want to see structures of these domains and so on. So this is, like I said, depending on the database. The second of the three databases that I would like to show you is PFAM. Okay, and it works very much the same. So they do the profiles and they allow you to search your protein against those profiles. And so what I want to show you is why this redundancy of databases and tools sometimes is useful. So if I run the same sequence that I used before in PFAM a while ago, I have to say, the output would be like this. And we can compare with the output that I go from the other database. So you can see that there are some differences. What is important here, so why this will be? Well, maybe this database PFAM didn't generate a profile for this domain yet, so it doesn't find it because it doesn't have it. This domain is like this one. I cannot see, they are using a different coloring. So, but I can tell you that it's the same profile. This one is the same as this one. And here we can read FN3 and they find some, but some of them they don't find. So they have also different cutoffs for detection of those motifs. And so the results can change from database to database. And in this case, with this database, I obtain more domains and less with this one, but sometimes maybe the opposite. So if you are really, really interested in a particular protein, so it may be interesting to try several of these databases. And then more details about PFAM that they also, so the structures, they are linked to Wikipedia, so that if you modify the annotations of in Wikipedia of this domain, then it gets transferred to the database and vice versa. Okay. The same thing, they have the structures, and also PFAM shows the distribution of the domain in different species using this type of visualization. I mean, if you have time, you can go on and look at it, but basically it is telling us that whether a particular domain is used everywhere in the tree of life, or for example, it could be a domain that could be a specific of vertebrates or a specific of primates, or which I don't think is any exist, and so on. So it can help you a bit to understand also the evolution of particular domains. And then the third database that I wanted to introduce, this is the CDD. And without entering in a lot of detail, the output of this database is a bit more detailed in that. So again, I'm running the same sequence, Sorla from human, which has this VPS10 domain over here. So this would be the sequence, and these are the features that are found. And also this database CDD, it's pointing to some particular individual amino acids that would be, because of what we know of these domains, we would know that they have some functions, or is finding some motifs like this DXSDE motif, which are also present in your sequence, and it puts this information together. So different databases, different features, different profiles, so small differences, but if you put the results all together, more or less they give you a kind of a of a consensus of what your protein may be containing. Okay. Any questions, more or less? I hope I was not too quick. But the main message here is, again, there are different ways to do the same thing. And my favorite of these ones databases is PFAM, but it's going to be integrated anyway into another resource. So
why is your favorite PFAM if in this example it is the one missing the most information?
Thanks for this challenging question. Correct. It is in general, it tends to have more profiles than the others. So it's much more, I would say, comprehensive in that respect. I used to work with people working on SMART, so that's why I saw this example where they have more domains than PFAM, but maybe it's not fair after all. Okay, PFAM is very comprehensive and it has a very good output. In any case, you will see now a message when we access PFAM that is going to be integrated in something called Interpro, which actually put together like analysis from different databases like I did here before. But I thought it's easier if I saw it than if you see that into a web tool. So like we went step by step.
Do you have to align those all by hand, one by one, or is there a possibility, for example, in the browser where you can say align smart PFAM and CDD?
No, this I decided myself for this presentation some years ago. Interpro is a resource that is going to absorb PFAM. You will see this now when we use PFAM. This year we will use using PFAM, but this will be the last year because next year we will use Interpro. But it's not so. Interpro gives you directly a kind of a view where you have different outputs aligned, but it's kind of a bit complicated in the sense that it's not so clear like here. Like I say, you see all the domains here. It's all more mixed, but it's a bit technical. So that's why I'm just explaining you why we are still looking at PFAM this year. So let's move to the exercise. So now we go in exercise mode again, and I have my window here to do things, and you should try also. So we go in parallel. So we are going to it's going to be linked to what we did before because we are going to use the same protein, but now we start with the sequence. So the first thing I want to show you is how we can quickly find structures of, for example, human myosin 10 in the PDB or of homologs, which can be helpful in some situations. Okay, and so for this, we are going to do a sequence similarity search. So what we are going to do is to take a protein sequence, which you don't need the sequence. We will use this identifier here. I think you can copy it from the script, or you can type it. It's Q9HD67. It's an identifier of human myosin 10 in UniProt. We had an identifier for the PDB structure, but we have another identifier in the sequence database. So we are going to we have to go to the BLAST from NCBI. I think you have been using BLAST with nucleotide sequences, correct? So now yes. we are going to, I don't know if you get to this window, you need the protein BLAST, not the nucleotide or these ones, but the protein. You should see BLAST P here. This is very important. I mean, you can always change if you want, but we need BLAST P because we are going to compare. What we are going to do is to compare the protein sequence, okay, the sequence of human myosin 10 against the sequences of all the proteins for which, okay, the proteins for which we have the structures in the PDB. Okay, so our human myosin 10 we are going to compare it against protein sequences, but those will be protein sequences of structures that we have in the PDB. Okay, because I want to find if human myosin 10 is already at least a fragment of it, if it is in the PDB already or not. We know because we have been looking already at one, but are there more? Which parts? And so I think you can follow the script. You will have to type the identifier. I go to the next step. So this would be the window you have, +P, remember? So you will have to paste the UniProt identifier here, and you have to tell the program that you are searching against the PDB. Okay, so you need to say here, the option is protein data bank.
And then blast?
And yes, please, yes. And then you, yes.
Can you show the UniProt identifier, please, again?
Yes, of course. Yeah. I need it myself, actually. 9H67. Or you could copy it from the, if you have the PDF of the script, maybe it's better to. Yeah, I'm sorry. So so you have there the thing, and then here is the option, and you have many. The default is good if you want to find homologous proteins, but we want to find if myosin 10 human has structures in the PDB, because you have a protein sequence and then you want to know more, and then okay, so which do I have proteins already? Do I have structures for this? And then you have to go here. I hope that by now everybody. A funny thing of bioinformatics is that the server of the NIH could be down, and we will have to find an alternative. So this is a lot of fun. So, but it tends to be okay, and it seems to be working now. Okay, so I hope you go already. So so you have to look now. You have a tab in the output. I put it here. So here you have a tab for the alignments. Okay, so you hit that, and then so you have now the hits. Okay, so the first hit. So again, sorry about going forward and backwards. If I go to descriptions, I have a list of the hits. Okay, and here it's telling me you can. I mean, percentage identities is good for you to see. Okay. So here very high, and then it goes lower and lower. So the list is very long, but we are going to focus on the top cases. But I would like you to look at the alignments, and then if the structure is known, okay, of the query, actually, you will have a link to the structure. You could click there, and then this server will show you. Okay. So in this case, we have a structure. It's not the one we have been using. Okay. Whoa. Why is that? It's different. Okay. Why that could be? It's myosin 10, but I have this thing here, and where is the chain B and A? Why is different? Okay. So why is different? What are we having? This is the question. So what can you someone can explain me what do the first three hits represent? And the question is, which part of the query was matched in our search, and which protein? I think we know. Okay, so if you a bit of a help, if you are looking at an alignment, the query is the protein we use to search. The query was the entire human myosin 10, the sequence. Okay, this is a protein that has 2000 amino acids, something like this. The subject, we have, for each alignment, we have one different subject. So here, this is the first hit. This is the second hit. My query is the same all the time, but the subject is different. The subject is now is defined. The subject is defined in the heading. Okay, this was one PDB, chain A of some PDB. The second subject is another PDB, the chain A. Third subject, another PDB, chain A. The query is always the same, and the numbers are important. So it's telling me this number here is telling me that the alignment, the similarity between my query and the subject starts, and with this I'm answering the question, it starts at amino acid 3 and ends at amino acid 776. Okay. So, and then also something just in case, the line in the center. So you have the query, the subject, and a line in the center. I'm going to go low in this list of hits quickly so that we see something a bit different. So for example, here I have my query is always the same. I have a subject that is something, is here, and then I can read the similarities and the identities between the query and the subject in the line in the middle. So in this alignment, where I have the same residue, the middle is showing me the residue. And if I have similar residues, the middle lane is showing me a plus. Okay, so I have things that are identical, and I have things that are kind of similar in their properties. Okay, so for example, a leucine is similar because it's hydrophobic to an isoleucine. Okay. So here, my query is not exactly the same as the subject, and that's why I don't have all identities. But what's happening here? Someone can, maybe someone can start answering the question before I say everything. So what can you interpret the first three hits? Which part of your query was matched, and which protein was found?
Maybe it's the myoglobin of a human and the others of
the myosin. Yes.
Yes, the myosin of human and the others of a mouse, maybe.
Oh, that's coming, but not 1, 2, 3 is 100% identical. So it's 1, 2, 3. Yes, so we it's 100% identical. So it's actually human myosin 10 in the PDB, and we have several PDB structures. Okay, because different people make different structure resolutions of the same protein. For example, could be different fragments of human myosin 10, or could be different splice variants, or could be mutants of, you could have the structure of a mutant of myosin 10, or it could be myosin 10 even if it was the same fragment, it could be in complex with different things. It could be myosin 10 in complex with this chain B that we saw before, a peptide, or in complex with another protein, or in complex with DNA, which I don't think it ever interacts with DNA. Okay, so you for the same protein, you can have different entries in the PDB, multiple ones, and that's why we see in this case 1, and we know it's this protein because it's 100% identical. So possibly, and also, also there is information up here saying Homo sapiens, for example. And so what is so if someone can quickly tell me which region is matched in the query. So which part of our query, because remember, PDB files, they may contain the structure of fragments of a protein, and our protein is very large. It has 2,200 amino acids. I could reverse the question. Do you think there was, is there a PDB entry that contains the structure of the complete protein? Do you think so? Okay, I think I will answer this question quickly. So if you look at the alignment, here we can see that the alignment starts in 3 and ends in 776. So we have a fragment, that fragment of human myosin 10 in this particular PDB in 5I101. Okay. So what about the fourth hit? So what is that?
I think it's very shorter.
Let's see. So this is the, sorry, I have to count. 1, 2, 3, 4. Correct, and which we can read it almost from the screen. So which part of the query is matched in this case? You have to read. Someone read the numbers, or I do it. So the values, you can see the values here. So it is matching my query in a different part. Before we have all the first 3 hits, they were matching human myosin 10 from 1 to 770. Okay, but now we have this match here, which, like your colleague say, is shorter. Is also human myosin 10, but it's going from 1486 to almost the end of the protein. So this is a different fragment of the protein. And if you would like to know something about this part of the protein, about residue 2000, for example, these PDB files would not help you because those are a different fragment in the N terminal. Okay, so you would like to focus on this one. And actually, if you look at all the hits, there is no hit against the middle part of the protein. So sadly, if you would be interested in the structure of the middle of human myosin 10 between 725 and 1400, you would find that there is no PDB there is no structure solved in the database for that part. Okay.
Where can I see how long my subject is? Because
this, yes, yeah. I think yes. Okay. So this is the start of the page, and it has there is a place where it says query length, and it says 2058. Okay, which I find, is that correct? Yes, yes, correct. Yeah. So for example, this hit goes up to 2045, so almost until the end. Okay. Yeah. And just to move on, so if so, there are some hits. If you go down the list, there are hits to proteins that are not human myosin 10, and then you may ask, maybe so this we will see on Monday. Imagine you would have, if the protein of the mouse would be very similar to the human one, and this we will discuss on Monday. The structure of the mouse protein could be helpful for you to know about the human protein because they are so similar, and then but it's not the case. So you have like I said, so we are missing the 700 to 1400, and so for example we have the hit to mouse is also hitting from 62 to 773, so it's the same N-terminal part, and we can go down and down and down to things that are less and less similar, and we will not find matches to that central part. Okay, plus the identities go lower and lower and lower. Okay, so you would need for that middle part, you would need to predict the structure. And this we will discuss on Monday, how this is possible. So for the next 20 minutes, just to finish, I would like to connect the domains of human myosin 10 with the structure that we saw in Chimera. So what basically this exercise now first is to find the domains in PFAM for this sequence, and then to color the last three domains, which are the ones in the PDB that we saw before in the previous lesson, to color the structure of in the PDB according to the predictions from PFAM. So remember that PFAM is doing predictions of where the domain goes. So with this, you can see how and if these predictions fit or not the real structure. You know, the predictions are useful if you don't have the structure, even sometimes if you have the structure, but they may not be perfect. This is something we have to take always into account, but they tend to be helpful. So I would like you to follow so this is the last 20 minutes, to follow the instructions and say, so first, if you search PFAM, I mean, if you Google it, basically you will find it. And then I'm going to cover again, sorry. So this is the PFAM page with a warning that is terminating and it will be available somewhere else soon. And then you have to search again. We are going to search a sequence, and you don't need to paste the sequence. You can just use this UniProt identifier there to see the domains in PFAM. Okay, so please do that, and then we will have a bit of time to discuss the output.
Quick question: which one is the one with like the actual data from crystallography and stuff, and which one is doing the predictions?
All of the ones I'm going to show you are all predicting.
Okay.
Okay, they don't have a they don't use the real structure to do that. But you will see that the predictions are relatively accurate up to a point. That's something I wanted to discuss. So let's see, this part in principle should not be very difficult. So you get the output, and then if remember the numbers that we were obtaining before. So in the initial or let's say we saw basically two types of matches for the region from 1 to 4,000. No, sorry, I don't remember. From 1 to 700, and then matches that were from 1,400 to the end of the protein. So if you have this output, you have here the colors, and here you have something more detailed. Okay, but then the domains is what is important for in this case. So you have to pay attention to this column, and you have the start and end predicted in your sequence. Okay, and then so the first. So remember that I told you that is if you are going to predict. Sorry, if you are going to resolve structures experimentally, sometimes you cannot run you cannot make X-ray or NMR of the entire protein. You need a fragment, but I mean, at least you need to cut the protein between domains, because the problem is that because domains are folding units, if you cut a domain in half, it may be that the half that is left is not folding properly, and this may form aggregates. Like imagine you have you have cut in the middle of you have a beta sheet that is composed of several beta strands. And maybe if you cut your domain and you have only two strands, maybe those strands cannot fall properly because they are missing the other two. And then they may be exposed hydrophobic residues, and those hydrophobic residues may aggregate, and then you are trying to solve the structure of this protein, and you cannot do crystals because the protein is forming aggregates. And this is valid for many types of research, of course. If you want to, if you want to know, what would be the function of my protein if I take out the DNA binding domain, then you need to know where the DNA binding domain is. So this is it's important to know where the domains start and end. And that's why in the previous exercise, many structures were obtained actually from 1 to 1,430 something. And if you look in the table, you can see that there is actually more or less there is like a that's like the second pH domain. So they would be no, sorry, I'm making the mistake again. Sorry, from 1 to 770. Sorry. So it's like ending those structures would have this HA3 domain, the myosin head, some of these domains, and then they will end up approximately after the second EQ domain. Okay, so it will end up over here. And then we have the other one, the one that we were actually looking at in the previous lesson, and that's starting in 1,400 something. And that would be after this pH domain, and it will contain these three domains in the end. Okay, so this is the one you have been looking at in the first lesson, the structure. Okay. So as a last part, then what I would like you to do, again, we get back to Chimera. You get the 3P set D. Should not be very difficult because you will have it. Okay, if I close here. So you will have it here. So if you click there, you will get it. And I remember, I'm going to, sorry. PFAM. So this is this fragment. It's the structure of this fragment of human myosin 10 with these three domains. And I would like you to don't close this PFAM window. Use these coordinates now to color according to any colors you want. I mean, orange, pink, brown, but you can red, green, blue, whatever you want. Of course, you are free to do that. Color now these positions accordingly in the structure, because I want you to, the predictions, they don't know the structure. They try to predict, but are they accurate or not? But we can see how accurate they are because we can see which parts of the sequence are matched here. Okay. So what I want you to do is to use these coordinates to color these three domains in Chimera, and then we will discuss that to finish the lesson today. Okay.
At which chain are those?
That will be chain A.
Chain A, thank you.
The B was the small one. In this case, we have only two chains. Some PDB files are very, very complicated. They will have lots of chains, and we will see maybe on Monday, you can actually select chain A, and then you say atoms, say delete, and then you are working only with B, right? And so on. So sometimes you have to hide some of those because it's very complicated. We have been looking at PDB. This PDB file is relatively small, but there are huge ones that are really gigantic. So please, you use the numbers you get in PFAM.
I think I'm done.
Okay. Shall we wait? I would say 3 minutes.
Can you repeat which lanes we should mark in the colors? So just the first from 7 to 767.
You need to open in Chimera this structure, and then I would like you to color these domains. MY myth, I don't know how to say this thing, myth 4 in any color. You do purple.
Okay.
RAS associated in orange and firm in pink or any other colors. In fact, PFAM has changed now, and I mean, I took these colors from a previous run in PFAM, and now PFAM gives orange. Sorry, gives orange to this one and pink to this one and brown to this one, but it's not. Obviously, that's a consensus you can do. So yeah, I wait 2 minutes more and then discuss. I don't know if maybe the person that was done could share the screen.
Sure.
I think, yes, yeah. Then I will, because I'm tired of sharing my screen. Yeah, perfect. Oh, I don't need to stop sharing for you to, or no? Okay, good. But this is interesting. So even if I don't stop sharing, you can share your screen. That's good. Okay, so everybody's seeing the structure.
Yes.
Okay, good. Okay, this is nice. I didn't know that this was possible. So anybody can basically take the screen away from the presenter. You are nice because you are not. You could be taking it away all the time from me, but then I will notice. This. Okay, so you can read the questions in the script. So, and I think that would be okay. So you don't need to see my presentation. So we can use this this screen now to you cannot, but I can do the questions for you. You don't need to.
Sure.
The person that is sharing, yes, yeah, don't stress yourself. So so the general question: how do the PFAM annotations that you have marked in this structure fit the real structure? Remember, these are predictions. And so what do you think? So
well, we have
know
seen that the actual protein is way bigger, and we have only a small part of the protein and just 3 domains. So this is not the whole myoglobin
exactly
protein.
so one first reflection is correct that that more domains could be defined for this protein. So this would require some manual work from the people doing these databases, which is always they have limited time, but it's good to ask them if you need it. They would have to take, for example, parts of this white, the white part that is not annotated. They could take it. They could collect more examples from homologous proteins, maybe from the mouse protein that was relatively similar. There was a Dictyostelium discoideum homolog, so that could be useful. They would align those fragments corresponding to the white region that looks like a domain. They would make a profile. They would search that against all the proteins in the database, which are millions, as we will see on Monday. And then with this profile, they could now search cases of this domain fitting the consensus of the little examples, the few examples taken, to then use that to identify now the domain in sequence in the new searches, basically. Okay, but because they don't have a profile yet for that domain, then that's why it remains wide. So what would you say about the borders of the domains? Are they well predicted? Because in principle, we would not like secondary structural elements cut in half, basically, because they could aggregate. So what do you think? You can zoom in maybe also a bit if you want with the exactly correct. Yeah. Look at that in the beta. Look at the beta strand. So there, for example, we have a beta sheet composed of 1, 2, 3 strands, and one is white. and no way it's clearly, and you can see, don't move now, don't move. Oh, sorry. There, perfect. You can see that the start of the next domain in red, it's quite okay. So you can see the jump, like I said, there's the alpha carbon trace is jumping from one domain to the next, and the red is starting quite fine. But we should add the white part should be added, is missing from the pink part. It's really, and there is no way that's not part of the domain. So all the beta strands that that compose a beta sheet are part of a folding unit. They cannot be cut away. Okay. There was another question maybe related to the white part. So which part of human myosin 10 is interacting with, remember there was this chain B here. So can you color the chain B in blue, for example? You have to select chain B, actions, color, whatever, hot pink, anything. Yeah, exactly. So the question is exactly, so which, so this domain that is not annotated is actually maybe important for protein interaction, because it's the one that is in fact interacting with the blue peptide, whereas the other three domains are not. So maybe it would be interesting to make a profile for that domain, to find it in myosin 10 and in other proteins, because this would tell us, this part of my protein is interacting with another protein, and so on. So you could learn from that. And I think we did quite well with this lesson. We are on time, so I would finish for the day. Any questions about this part or anything? I'm going to stop the recording before I forget. And
yeah, can we maybe get